Hello, everybody. My name is Ghassan Abou-Alfa from Memorial Sloan Kettering Cancer Center in New York. It's a great delight on behalf of all of my co-authors to fill you in and update you on the presentation that we had at GI ASCO 2024 in San Francisco, California. In that meeting, we were honored to present the final results of the Proof 301 study that looked into the phase 3 clinical trial of the oral FGFR2 inhibitor, Infigratinib, versus Gemcitabine plus Cisplatin as a first-line treatment in patients with advanced cholangiocarcinoma or intrahepatic cholangiocarcinoma. Of course, for the patients only with the FGFR2 fusion rearrangement. The study was to randomize patients and was planning for a rather large study looking at overall survival as a primary endpoint. And as you can imagine, this would require, as we mentioned, a prior screening of the patients for the FGFR2 fusion. We ended up screening to close to about 1,100 plus patients. And this is over close to about a tad less than 4 years. And if anything, we could not ultimately enroll except 48 patients. Why is that? Because the alteration is rare to begin with. And of course, we had a randomization that would, of course, not necessarily place everybody on the Infigratinib arm. What did we come up as conclusions from that study? Number one is that no doubt that we cannot answer to the questions that we wanted to ask to begin with, being overall survival and progression-free survival and the response, because the numbers were very limited. Nonetheless, especially in regard to the responses, were commensurate with what we have seen beforehand in regard to Infigratinib and what it can do. The second thing that we were able to conclude is that from the adverse events, understandably, they were there, they were appreciated, especially the ones that we are well aware of, being elevated in regard to liver function tests, the Hatfield syndrome, the neutropenia, and of course, these were within the confines of what we would have expected from the drug to begin with. The hypophosphatemia that we already reported quite a bit on in the prior work definitely was noted as well, but thankfully it was really appreciated further, and we knew now to ensure that we provided with the appropriate care, but not overtreated so we don't turn it into a hypophosphatemia. However, as you asked and wondering, still, why it didn't work? This was one of the key aims of us reporting that data, because it's a rare malignancy with a rare genetic alteration, and we're trying to really do a large clinical trial to find out an answer in a randomized phase 3 clinical trial. Realistically, this will have taken long time, enough that it will not really be feasible from the aim to get the therapy available for patients. Number two, it will not be feasible from the academic evolution, how studies do evolve. And of course, financially, it will be a stressor for whoever is supporting that study. For that reason, we do understand the sponsor elected to turn off the accrual, elected to disabandon the study, and also elected to pull the asset of the drug itself, Infigratinib, from any further development and further applications. We respectfully accepted what the sponsor have the right to decide, but at the same time, we did agree with the sponsor that we still are obligated to assure that our patients and loved ones and the whole public do understand what we have done in that study and why we failed. If anything, the failure, we thought it will be a good invitation to, number one, proudly always, no matter what the results are, this is what's nice about science, you have to report it, and we have all to report it. Number two is, we hope that this will be an invitation for the agencies, wherever they are, including the US agencies, to appreciate that patients with a genetic alteration that's rare to begin with in a rare malignancy we will not probably be able to assess and finalize data based on a phase 3 clinical trial with a primary endpoint of overall survival, and we might have to probably accept settling to a phase 2 clinical trial looking into indirect asset results like response, progression-free survival, translating into hopefully improvement in survival. Because here, what we have seen, we sadly lost a great asset, being the Infigratinib. It's not available anymore. And we hope that we'll be able to reverse that for not only this drug, but for many others. We thank the sponsor for supporting us on that effort, and we're proud that we did report it, and no doubt that we are also working on the final publication for that same purpose. We elected to proceed with presenting that data, despite that we could not accrue the study, because we felt an obligation that a promising efficacy already noted in a phase 2 clinical trial for the Infigratinib. Number two is a big excitement about the anti-FGFR2s in the whole scene of therapy for intrahepatic cholangiocarcinoma. But the fact that we could not accrue the study as we wish to. And number two is because we feel obligated to report to our patients who were part of the study as well as the loved ones, and of course an obligation to the whole public about why the study did not reach its goals. So for that reason, on behalf of all of my colleagues, we did present the final results of the Proof 301 study. We invite patients always to make sure they have the next generation sequencing of their tumors as soon as possible. And we encourage all physicians to support their patients getting the next generation sequencing. Understandably, this is still not there yet in regard to get the responses immediately. The challenge of getting the tissue, challenge of doing what's processes needed to prepare the tissue and ultimately get the results. And on the average has been really optimistically 3 to 4 weeks and realistically 6 to 8 weeks. So no doubt that to bring this all into first-line therapy might be a little bit more difficult. We hope in the future we'll have a more prompt way of getting the genetic alterations answered immediately rather than await for the results and as such deny the patients any options of therapy. So one might ask, like, then what? Okay, we lost that asset, but then what are we going to do with our patients with the and FGFR2 genetic alteration fusion? The good news is that thankfully there are other drugs that are available, among which are pemigatinib, which already are FDA approved conditionally because of phase 2 trial. The futibatinib, also same thing, and they are readily available for patients. And we hope that they will get full approval rather than wait for a large phase 2 trial, a phase 3 clinical trial. Important also to remember that a big challenge still we have is that patients might not necessarily continue benefiting from therapy, and they will probably have resistance to certain therapy. What shall we do with those patients? This is where proudly we are bringing in the novel approach that these alterations, after all, are deterministic. They are not really by random and endless in number. And as such, we're trying to see how patients do circulate from one therapy to the other, and maybe they can come back to the same therapy. Proudly, this work that we're currently ongoing, and hopefully will be within the context of that intrahepatic cholangiocarcinoma with the FGFR2 fusion to give more answers for the future. I thank you again for listening. As we said, this is a Proof 301 study looking into Infigratinib versus Gemcitabine plus Cisplatin. We are proud that we were able to achieve the work. We're proud that we were able to report the work despite that we could not finish it. And the reason is because of the poor accrual, because of the challenges of a rare malignancy with rare genetic alteration to be given in first-line therapy. And I hope this will be a learning lesson for all of us so we can see how we can move forward the field with novel approaches, how to assess those therapies and not await for overall survival phase clinical data. I thank you for listening. You have a good day.